It's a pleasure to welcome you all to this Krishnaya lecture. It's an honor for me to introduce Katherine Roeder, who is a UPMC Professor of Statistics and Life Sciences in the Department of Statistics and Data Science and Computational Biology at Carnegie Mellon University. She's developed statistical and machine learning methods in a wide spectrum of areas, including high-dimensional data problems in genetics and genomics. She's one of the leaders of the Autism Sequencing Consortium, which is an international organization dedicated to discovering the genetic etiology of autism. She's been developing statistical methods for the analysis of single-cell RNA-seq data, especially brain cells, and for the analysis of single-cell CRISPR pooled screens to evaluate the impact of precisely targeted perturbations on the whole transcriptome. She's received a long list of awards, so I'm not going to go through all that long list of awards, except to single out the fact that in 2019, she was elected to the National Academy of Sciences, which for statistics for being as important it is to all disciplines of science, the number of statisticians who are in the National Academy of Sciences is still relatively small. That's quite an honor. And of course, my favorite thing to tell you about Katherine is that she's a 1988 PhD in statistics from our own university. Her thesis title, in case you're curious, is Method of Spacings for Semi-Parametric Inference, and her advisor was the great Bruce Lindsay, who many of us had the honor of knowing for many years. She came in, I talked to, there are only a few faculty members I know who knew Katherine well as a student, and what I heard from them was that she was brilliant, and she came in with already a very strong interest in biology and genetics, having had her bachelor's degree in wildlife resources. So with that, I'd like to, so before I present the plaque for a lectureship, I just want to point out that this Krishnaya lectureship is really quite a distinguished lectureship. The list of people are all names you'll recognize: Brad Efron, Jim Berger, Nancy Reed, Elizabeth Thompson, every name, really a who's who of statistics. This is the first time we've given it to one of our own alum. So with that, Katherine Roeder.
Thank you.
So I should read the citation.
Thank you.
Professor Katherine Roeder is selected as the PR Krishnaya Lecturer for her wide-ranging contributions to statistics and machine learning, for developing methods for high-dimensional data problems in genetics and genomics, and identifying genetic factors for autism and other psychiatric disorders. With that, congratulations to Katherine, and welcome to back to Penn State.
Thank you.
We will have a question and answer session at the end of our talk, where you see the QR code up here, so you can use your phones to enter questions. Your phones are your laptops if you're carrying your laptops. And you can ask questions which will be curated by our grad students here who have done this before and done it very well. So Caitlin and Wangu have kindly agreed to run the Q&A session. So we will, right after the talk, we will have a Q&A session that's run by the two of them. So with that.
Thank you. Thank you so much for this great honor. When I looked at the list of previous recipients, I was definitely humbled. But I did not know Krishnaya, so I thought instead I would tell a little bit of a story about my early days at Penn State, which I'm sure are different than what other people remember. So I never meant to be a statistician. I had studied to be a wildlife biologist. That was my intention. And I had a bad skiing accident, and that was not really a possibility. And one of my professors said, well, I went to Penn State and studied statistics. You can do it too. So, but the thing is, I had no qualifications. I might be the least qualified person that has ever been admitted here, and but nevertheless, they did admit me, and I came very enthusiastically. And the first day, you know, the group of us who had been admitted that year were in like a little seminar room, and Bill Harkness, who had been the chief for apparently decades, he was giving us our introductory speech, you know, get us all started for the year. And he said, I'm really excited to have you join our department. And he looked around and he said, well, I'm excited about some of you. And I thought, he means me, and not in a good way. but I was never offended. I thought, I like that guy. He's saying what he thinks. We're going to get along fine. And I'm still immensely grateful that Penn State gave me the chance to, you know, to make up what I was missing in my background and to succeed and have this opportunity to give this lecture so many years later. I just only wish Dr. Harkness could be here. But I do have 2 Jims here, so that's good. Okay, so let me get started.
Sorry. I'm wondering if your mic is on.
Oh, are you guys having trouble hearing me? Are you able to hear me, though?
It's only for recording.
But the question is, can people in the back hear me? Yes, okay. Okay, great. Okay, so at any rate, I did stay with my interest in genetics, and I'll talk about some of those topics today. And let me explain what all these words mean that you might not know in the title. So first of all, CRISPR is the most exciting development in genetics of the decade. And the idea is that you can make these small changes in the genome, and this will lead ultimately to the ability to repair errors in people's genomes. It's extremely exciting area. And the kind of topic I'm going to, the kind of data I'm going to talk about is blood cells, or particularly the immune cells. And so this is just a picture of the fact that if I took a sample of your blood, I would get all these different kinds of cells, and in particular like the T cells. And these are the things that would fight COVID if you were attacked by it. And so, but each cell has a different job, and that's what's so exciting about the single cell part, is that you can measure the different kinds of cells rather than just blood in a mixture. And so just to give you a sense, this is not my talk, this is just what people are doing with CRISPR. So they would take a person with cancer and take out their T cells, and then they would make these little snips and design these the genome a little different in a handful of genes, and then they would put it back into the person, and those cells would fight and kill the cancer cells. Now that is just too cool for words. It seems like science fiction. But somebody had to do the research that decided what were the genes to change. And that's what my research is about, that kind of thing. If I change this gene, what does it do everywhere else? Okay, so just now, what is this about? You know, we all have the same genes. There's no good and bad genes, but in every cell, different genes are talking. And so the way that we know which genes are active is we look at the RNA and we count how many copies there are. And then we can know which genes are active. And so if I knock out something in your DNA and that gene stops acting, then I've learned how the mechanism works. And so anyway, now you want to know, because you're statisticians, you think that's about enough biology for me. Let's get to what's the data look like. And so, you know, every time I give a talk, I think, how can I find a good statistics picture of single cell data and data structure? And my postdoc said, let's ask ChatGPT to make a picture. So we did. And this is what it gave us. Now, I don't know, is that single cell or is that a bathroom tile? But we're going to make it into single cell. So every row, so that's a matrix, right? And the color, ignore the different colors. ChatGPT always gives you what you asked for and a little more. Suppose it was monochrome, black and white, and the more intense the color, that's the bigger the entry. But first, the rows are cells, and the columns are genes. So I have many, many thousands of genes, and at least hundreds, if not thousands, of cells. And the white entries are zeros. And in real single cell, what makes it so hard is it's so sparse. 95% of that matrix would be zero. But nevertheless, we want to see when are different genes turn off and on, because even though they're mostly zero, they're not all zero, and that's where the information comes. Okay, so lots of zeros. And so what my kind of experiment, I say my, but I mean the scientist I'm working with, is they're going to get these single cell CRISPR screen readouts. In other words, they're going to get these matrices like that bathroom tile, and they're going to try to do it to understand the function of certain things, and they're going to use CRISPR to change something. So we're going to go in there and break something, and then we'll look at all the genes and see what they do. So I can do some cells that were the control and some that had the CRISPR change, and we want to compare the perturbed and the non-perturbed cells gene by gene. Okay, so this is again what it looks like. Let's say I have three things that I want to know. This perturbation, say this is killing one gene, killing a different gene, killing a third gene. And I'm going to put these are cells. Some of the cells get each kind, never more than one. And some of the genes have that NT, which is like the placebo. So something got into them, but it means to do nothing. So now I'm going to get a machine that's going to spit out all the gene counts. So I'm going to have those counts, and I'm going to say, for each perturbation, did it affect this gene, that gene, that gene, next perturbation? I'm going to do these all in pairs, one at a time, like this pair and that pair. So it's pretty simple. It's incredibly simple. There's no reason why this wasn't already solved, but it wasn't solved well. Okay, so this is what we're doing. We want to know what perturbations impact what genes. And so, again, this is what the data looks like. So I want to test first the blue ones compared to the controls. And for each one of them, remember, for every cell I get, for a gene, I get a gene expression. So this is a fancy looking violin plot to show that these distributions are not nice. But there's the distribution across all the cells there. And you can ask, is it different in the blue versus the gray? Just a two-sample test. I get a p-value for every gene, and I want to find the genes that are significant, small p-values. That's the whole story. Very simple. Any questions? Okay. So anyway, let me give you a little bit of notation. Let's let X be the gene expression. That's a count variable. It can be many, it can be 0 up to a large number, generally small counts. So we usually model that. If we're going to model it, we'll model it with a negative binomial, because it's a small count like a Poisson, but we have some over dispersion, so we use negative binomial. X is the binary, and Y. X is whether it was treated or not, Y is the small count. And the Z is also important because in every experiment there's various technical factors. What technician did the experiment, what batch was it, what temperature, those kind of things. And the worry is that sometimes these technical factors are confounders, and And then we have to adjust for these confounders, and that's a little bit of a worry. But we have measured them. Okay. So now here's finally our test. I've got one gene, I got one perturbation. Are they independent, conditional on the technical factors? That's all we want to do. And we want to have a well-calibrated test so that we don't get type 1 errors. And we're going to apply this test hundreds of thousands of times for different gene perturbation pairs. So there are many, many tests, but each test is just a 2-sample test. Okay, so now, well, there's of course things in the literature. I'll just talk about these first 2 easy ones. There's a Wilcoxon test, which, as you remember, it's a rank test. And there's a negative binomial regression. Let me go back to my notation. So for the Wilcoxon test, can't find my mouse. Oh, okay, for the Wilcoxon test, I'm ignoring Z, and I'm just looking at the ranks of the Ys in the cases versus the ranks in the perturbed, the not perturbed, the case, the control. And that's all I'm doing. If I'm doing the negative binomial, I can do a regression. I can regress the counts using a negative binomial. And this is my most important covariate. I want to look at the beta that goes with that. But I can also control for these. Very, very simple. They certainly should work. How could they not work? They taught you that they would work. Okay, but let's see if they do. Okay. So, and there's some other tests too, but I'm not going to talk about them in detail. So anyway, this is just a picture that I pulled off the web to explain, because in people in genomics and genetics, they use these plots all the time, but I learned on Wednesday when I gave this talk to somebody else, statisticians aren't always familiar. So let me explain this. Okay, I think I forgot to tell you something. I am going to apply this, I'm going to apply my test in a case. I'm going to first look at the calibration. I'm going to do controls versus controls, because I want to see if I'm getting false positives. So controls versus controls, every p-value, there's a p-value for every perturbation in every gene, lots of p-values, but they all should follow the uniform distribution. So I'm going to do a QQ plot, but the QQ plot looks different than you're used to, and this is how we always do it. We always take the observed and the expected, and then we convert them to the minus log base 10. So like an 8 is a p-value of 10 to the minus 8th. Okay, so the ones up here are the potentially significant ones, the smallest p-values. But this, which I got off the web, is perfect because all of the tests fall right along the line. There are no false positives here. I mean, up here they come up a little bit, but those are not significant given the number of tests. Okay, so that's what the data looks like. Now let's look. Remember, I had a bunch of tests off the shelf. Let's see how they did. And this is on negative control data. They all should go along the line. And this test is horrible, but even the best tests aren't calibrated quite right. So the first thing we did was we said, why are those tests not calibrated right? They should have been. The Wilcoxon is non-parametric. The negative binomial, that's what we in the single cell world always use. What went wrong here? Okay, so once again, to make sure you got it, they're wrong because these things are arching up and many of them are significant, and that would be a false significance. Okay, so anyway, what are the problems? The first one is the sparsity, because the Wilcoxon rank test is a pretty miserable test when all of the data are 0. 95% of the data are 0. That's a lot of ties. Okay, so we come up with a measure of what's the effective sample size, and we count how many non-zeros do we get in the treatment case? How many non-zeros? And that can be, you might have had hundreds of samples, but maybe only 12 of them are non-zero. That's the effective sample size. That's where the information is. And then there's confounding, and of course the Wilcoxon can't take the confounding out. The negative binomial can, but the negative binomial might be the wrong model. We might estimate the dispersion wrong, or we might model the confounders wrong. So these are the things that can go wrong. So we start to look at those. So first,
Clarification question. Effective sample size, it needs to be non-zero in both the cases and the controls, in both.
well, all we do is just count. We just count if it's non-zero in the cases. It's just a simple ballpark estimate of what kind of information we have. It can be zero in the other one. Yeah. Okay, so now the Wilcoxon, we had great hopes for that it might work, and so we created tranches. So these are the worst ones, these purple ones. Effective sample size very small, and then these bright blue ones, turquoise ones, are the bigger sample size. And you can see the bigger sample size, it's fine right there along the line. This is the negative controls. But all these are false positives. So the effective sample size is the reason the Wilcoxon is not working. Now, why does that happen? It's because when you do the rank test, they call it non-parametric, but it's not completely non-parametric, right? Because once you get a z, you compare it to a standard normal. And you can see that when we took the bright blue, the big sample sizes, it is standard normal. But when we took the data from this tranche, it's not, it's lumpy, it's not normal. You're going to get a lot of false positives and just a mess. You're not going to have very good power either. So that's the problem there. Too small of sample. And yet, these experiments cost millions of dollars, like they don't want us to throw out. If you wanted to only have the bright blue data, you would have thrown away most of the data. So they're not going to let you do that. You got to try harder. Okay, so this is the confounding. And now, at first I thought, what's the matter with these experimentalists? Now let me explain why I thought that. They have different, they can't measure everything at once. They run out of room on their experimental platform. So they have different batches, R1, R2, and R3, and most of the cases were in R1. You say, what's the matter with those guys? Well, remember, they put those things in a test tube, and they randomly absorb or don't absorb, and by chance, it's uneven. And so batches are always different, and so you can see this pinky color has got a lower expression, so you're going to get a lot of false positives. You have to regress out the batch, but you can't very easily in the Wilcoxon, so that's a problem. Okay, what about this model misspecification? Quite honestly, I didn't think this would be a problem, but it is. This is a data set, a kind of famous data set, negative binomial giving lots of false positives. We've removed the confounding, we're doing the best we can, and we got a lot of false positives. So now we say, okay, now we know that things off the shelf are not working. Let's see if we can fix it. But the thing is, and this is what maybe statisticians don't realize, scientists want the simplest thing they can get that works. If it's beautiful and you really love it, they usually don't. So the simplest thing that you can get, because they can understand it, that's what we're aiming for here. Okay, so anyway, what we have, this is what our data looks like. Remember, this is the vector of counts. Each row is a cell. This is whether it's a perturbation or not. And these are our technical covariates. And the first thing we do is we just run that negative binomial and we get the z. We just do the best we can with that. And then we do what every scientist loves. we start permuting, and we get Z again and again and again under the null hypothesis, get the distribution, and no matter how lumpy it is, we can still get it, whoops, and so we can get the empirical p-value. And so that is our test. It's very, very simple. The real contribution here is that my marvelous student coded this up so that it's really fast. But that's what we do. And it's like amazing. It works. If there's no confounding, it's always valid. We call it scepter. It's always valid. And if there is confounding, it's valid pretty broadly. As long as the GLM is like approximately right, even if the overdispersion is wrong, if it's a decent sample size, then it's still correct. And so we can remove the confounding as long as the sample size is not too small. If it's too small, they have to throw it out. Okay. So, and you can see here that this is our test in the red in 2 different data sets right down the line, and there's the ones not working. So this was a very simple, I was asked to give a talk that showed some simple stuff working, and this simple stuff really did contribute. It'll change, you know, how scientists do this. And so anyway, my student I'm a terrible coder, but he's a good coder, and he coded this up to be very, very fast. And if anybody's interested, you can find it on my collaborator's GitHub. So that is the end of that story. Now I'm going to tell a more statistical story for a different thing to do with the data. But any questions before I'm just going to summarize that work, and then we can move on to the other. Okay, so the main thing is that when you want to write a paper that influences scientists, you have to use real data, and they're not really interested in simulations. And so we used real data, but we made sure that we knew the truth by doing the negative control. And these methods were fast, and most importantly, and this is the thing that I always struggle with, scientists will never use your package if you don't have good software. So we worked hard on that to make that something that would change the field and not just be a publication that dies. Okay, so now next problem. Again, same problem, not the. Remember, we've got the cells and the genes, and the expression is the intensity, and really what we're doing is we're comparing this mean to that mean if these are the control and those are the cases. So suppose we wanted to do something more interesting. Maybe this isn't very interesting, to test if there's any differences. So we might test the global null. That seems kind of extreme, but we could. And so we call this H0 global, which means every gene expression is equal. And let's see what happens if we try to test that. It's going to be challenging because there are too many genes. You know, like everybody in you know, we talk about high dimensional, and people are always talking about that, but in genomics, it's really true. P is orders of magnitude bigger than N a lot of times. So this is not going to be a good thing. It's not interesting either, but that's one thing. But there is a rich literature in the statistics for this. We go way back. We've got Hotelling's T-squared. We could do that. But that needs to have a small dimension, so that won't work. We don't have to go all the way back to 1932. Moving forward, people have done L2 kind of tests. So they just look and see if there's an accumulation of small signals. Or you could look to see if the L infinity is different. Those are possible things to do. Or we could look and see what scientists are interested in. Scientists are not interested. They never do any of these things, ever. So we don't want to go that direction. We want to say, what are they doing? Okay, so earlier, they are interested in the gene by gene, but if you can imagine, if you give somebody a list of like 317 genes out of a list of 20,000, it's hard to interpret, right? They want to know what's the biological meaning of those differences. What's going to be different if I knock this gene out and try to cure cancer? What's going to happen in a bigger set? So they want to know what biological processes are changed by this perturbation. What's the function that will change in the brain if I do this? What are the molecular pathways that are going to just act differently? If I could answer that, then they would use my methods. Okay, so of course, generally it's always true. Many people today ask me, how do you pick a problem to work on that is like relatively new and that people are interested in? And I will tell you right now, you find the thing they already published with an ad hoc method. They got an ad hoc method and then you try to make a proper method. You will never, never be first to get the ad hoc method. They always have one because they're not restricted like we are. Like we have these rules, you can't do that, but they don't, they'll do it. Okay, so this is, and these are statisticians. Steve Horvath is a good statistician. They made this package, and it's been cited tens of thousands of times. And what the package does is it says, remember I've got many, many cells, and for every cell I've got the expression on all the genes. And so I could get a correlation matrix, a gene-gene correlation matrix. Now this looks kind of pretty, and I'll tell you they messed with it. I mean, they didn't get 10,000 citations for doing a correlation matrix. So they tweaked it, and one of the things they did was they raised all the correlations to a power, and there's more to it than that. But when you raise the correlations to a power, they either race off to zero or they don't. And so, like if you raise the correlations like 6 power, you kind of get this nice structure. It's kind of cool. You find remember these are gene by gene, so these genes are working together, so to speak. And you got these correlations. You could test, instead of gene by gene, you could test that whole set. And so what they do is they take that block diagonal and they get the first principal component, they get the loadings, and they multiply by the genes, they get what they call an eigengene. Because the other secret to success is naming your stuff cool things, right? And then so an eigengene I feel very modern, and yet it sounds familiar to me. So they're gonna test these eigengenes. All right, so we think that's a good idea, but maybe we can do better. Okay, so the shortcomings of this is sometimes they call them modules, these clusters, and sometimes there's like 1,000 genes in there, that's too many. So the first thing we thought is we're gonna use sparse PCA so that we get cleaner, smaller modules that are tighter. And so we're gonna get sparse PCA, meaning our loading vector has maybe 20 positive elements and then all the rest are 0. And that's gonna be more interpretable. They might like that better. And so, first thing we do, I'm going to explain this data later, but this is my correlation matrix, and you can see that it definitely has some nice blocks in there, and those blocks are about a sample size of 20. And if you look over on the right, you'll see that I plotted the eigengenes for PC1 and PC4, and the colors are the case controls. And if I don't get significance there, then I'm kind of a loser, right? Because you can see that they're different. You can see they're different. So let's see what we get. So I'm going to test these. I'm going to talk about how to test these blocks. Okay. And so just to see it again, that's the first block, and you can see there's a good positive correlation for those. I think there's 18 genes, I can't remember, and those are the loadings. So all positive and relatively big for some of them. Sometimes I point at the screen so you guys can see me pointing at the screen there. Yeah, okay, so that's what I'm gonna do. I'm gonna build a test on that. And now instead of doing the global null, I'm now going to test the projected null. So see, the projected null is the difference in means, but I've got my sparse weighting vector there. So that's really this eigengene minus that eigengene, testing to see if it's 0. So V is the sparse vector, and we're going to get these from the correlation in the control cells. So I get V hat from that. And of course, we can prove that V hat converges to V. And so the question is, can we get a test? This would be like the numerator of our test, right? Is this going to make a good test? And so we get the sample means and the loading. And I would never be telling you this if this worked, because this is kind of easy. This is actually not going to work. Okay, so what happens? All right, so it's much more challenging. Even if we split the sample, in other words, we estimate V from part of the sample and the mu hats from another part, we use cross-fitting, no matter what we do, it doesn't work. And what happens is that we want this thing to be asymptotically standard normal when this is true, right? This is our hypothesis. We're not testing that. We don't think that's true. We're testing this. We want our test statistic to be standard normal then, but it is not. It is standard normal here, but that's so extreme. We don't want to test that. We want to test this. And so this is what happens, is the green one is in what we want to be standard normal, but we're obviously going to get a lot of false positives. It's only standard normal when this extreme case is true. And so we can't build a test on this. We have to fix it. Okay, I'm doing pretty good time-wise. Okay, so anyway, how do we fix this? The problem is that V hat, you don't really think about it that way, but it's a non-parametric thing, and those non-parametric things need more care. So that test I've been talking about so far, suddenly it's called T-simple. That's just the mu hats and the eigengene difference. That test ignores the fact that V hat converges poorly. So we have to do something else. And so what we're going to do is we're going to borrow something that comes from influence functions, which were really popular back when I was a student, but now they've become back into fashion. You know, I feel like I slept for 30 years, and then the same stuff I studied in graduate school is popular again. It's like great for me. So this de-biased one-step estimator means that you want to test for this. You want this theta hat test to go, you know, when you subtract the truth, to go to standard normal. And it's only going to happen if we put in this bias correction term. So, and there's a whole bunch of literature about how to do it. And because I was told that first-year students have to be able to understand this, I am not going to make you suffer to get this. I'm just going to show you a picture of the bias correction term. But it is, oh, you know, lots of papers written about how to do this. But this is the idea in a picture. So this is our T-simple. I need more stuff. Okay, this is the true distribution. If only we could calculate our things under the truth, then we can see that T simple is just bad. It's a long ways from this is what we're estimating, and this is our estimate. It's bad, far, far away from there. So could we get closer? We can only estimate in the sample, right? We can't estimate we can't know the true distribution. But just like with Newton-Raphson, when you take a guess, you can always take the tangent to this functional, always take the tangent, we get a little closer. So if we took the tangent and we estimated the tangent, we would get closer, and this is the tangent is the influence function of our estimator, which we can actually look up in the literature. So we get the tangent and we take the one step, and it is better. Now, it's not that good, but if I had a bigger sample, the tangent would be up here and it'd be pretty good, right? So these things work when the sample is big, not real big, but bigger. This one step will be quite a bit better, and that's why it works. And that's why when all of you students are going to see, you're going to just be browsing around and you're going to see the word debiasing next week. And you're going to say, hey, I know what that is now. That's just easy. It's just like a Newton-Raphson, only it's a Gateau derivative. Okay, so that's what you're going to do. And then voila, once we put it in there, the blue thing is the debiased one where we took that one step, and it's perfect, whereas this was the simple. So it really does work. Okay, so I got a couple minutes to go. Maybe 5, yeah. So I'll show you my example. So this is lupus is blood disease, and I've got cases who have lupus and controls who have lupus. And for all these people, I collect single cell, and I'm going to look. I got 4 major cell types. I look at those one at a time. So on the first, I'm going to look at the T cells, T4 cells. And I'm going to, for each individual, this is just for those single cell nerds, I'm going to get the pseudo bulk by adding up all their cells for each individual. So I've got 1.2 million cells, but I'm going to just add them up so I get a sample size of 169 and 99. And I can do a case control difference, and I can do these eigenvector kind of test. Okay, and I'm going to do that on 13,000 genes. So I've got a small sample and a big P, and it's still going to work. Okay, so I don't know the truth, so I don't know for sure it works. But remember, I'm going to test these sets of genes. I'm just going to test these 4. I could test more, but this is how many I could show you. And I'm expecting significance for these 2. So let's see what happens. Okay, now there's various things I could do. I could just do I could do a Poisson test. I know I'm going to get some false positives, but that'll just tell me where the significance is. You know, just do a regression. I could use Scepter, but that's too much trouble. I just want to do this simple thing. And then I'm going to skip the lasso in interest of time. That'll give me very, very sparse findings. And then I can test those top 4 PCs, which is what I promised to do. Okay, and this is what happens. How many of you have seen upset plots before? I'm like quite fond of upset plots. Mostly not. Let me explain these. These are the Venn diagrams of the future. So if you want to see a Venn diagram with 6 circles, right? So this is a Venn diagram. For the Poisson one, the ones that were Poisson only is this bar. The ones that were PC1 and Poisson are this bar, and so forth. So you can really see what's in a Venn diagram in a very nice way. It's called an upset plot. Okay, so anyway, pretty cool. You can see these 2 PCs are significant, barely, but significant and more significant. And PC1 is perfect. All of those 17 genes were also marginally significant. So we believe not only is the set significant, but each one is significant. Because when we test it, we only know that the set is significant. Now, let's look at PC4 and we say, 10 of them have a marginal signal too, but 9 of them don't, and there aren't any others. there's 19 in that set. What happened there? Now, when this is not wrong because the set is significant, but only 10 of the genes are. Then we can look at our sparse PCA and our correlation, and we see these are the 19 genes, and red is positive correlation and blue is negative, and we see that there's a set of tightly correlated genes and another set, these ones are correlated, and then there's this weird negative correlations. So what's going on there? And so I said to my postdoc, I said, you forgot to remove the mitochondrial genes, which we shouldn't have tested. So this whole set is genes that don't make any sense to be testing, and we take them out, and they shouldn't have been significant, and they weren't, and now we get this nice tight set, and those are the 10 that we found. So we feel pretty happy about this. It's interpretable, and so let me just wrap up. So the individual gene tests, they're very nice, they're important, but they're hard to interpret. The WGCNA module is very, very popular, and so we want to replace it with something that's more valid. You know, they also use the data twice, so they're getting those false positives, but nobody knows about it. And we're going to correct for that, and we're going to get the bias correction and get a valid test that answers this question, but is valid. And so I want to say one more thing, just for, you know, when you're doing your applied work, sometimes really simple things work and people are really happy with them. But if the simple things are wrong, then you have to do something else. And the PCA, because it's non-parametric, it does have some problems with it. And so let me just quickly acknowledge my collaborators. That second half, the postdoc is Tianyu Zhang, and he'll be on the job market, not this year, but next year. My longtime collaborator, Jing Lei. And then for the first project, Tim Barry is my wonderful graduate student who's gone on to Harvard as a postdoc, so he'll be on the market in a few years. And then my former postdoc, Gene Katsevich, who is now on the faculty at Wharton. So thank you very much.
All right. Okay. All right. And here, if you want to come stand actually in the middle between us, and that will be even easier. Okay.
I feel I'm in some danger.
All right, so hello everyone. My name is Caitlin and this is Juan. And so we'll be moderating the Q&A. So as a reminder, this is the Slido code that you can use to submit questions anonymously, and we'll ask them on your behalf. So for one of our first questions is, so from the first part of your talk, you were talking about the negative binomial and how that really wasn't working, model misspecification and things like that. At any point, did you ever examine possibly a zero-inflated negative binomial to also possibly help with the sparsity issue as well?
That's a very good question, and we did, and it didn't. Yes, the adding the extra zeros didn't seem to be the problem. I think the problem with the negative binomial is it's very, very difficult to estimate the over-dispersion factor. There's not much data for that, and you have to estimate a different one for every gene, so they tend to be poorly estimated. I think that's where the error is coming in. And remember, it doesn't get a lot of false positive. It's just a small number of the genes that are badly estimated.
And there's another question from the audience. So when you're talking about the correlation analysis, you use the one-step estimator. Mm-hmm. And why does the device distribution have a smaller variance rather than a shifted mean?
It's not that the variance is smaller, it's that there's a bias. So the bias In the end, it's like a bias squared, right?
Oh, sorry.
Yeah, sorry. Okay, so let me say that again. In fact, actually, can you guys can all hear the questions, can't you? Can't hear the questions, sorry. Okay, I'll say the question again. So the question was about the debiasing. It looked like debiasing made the variance smaller, but I don't think that's what happens. It's that the bias makes the statistic more variable. And so, it's like mean squared error. It's bias squared plus variance. So it's not that the variance is smaller. The variance is actually bigger, but once you divide by the proper variance and remove the bias, the whole thing converges. That's the difference.
So another question that came in is how long does it take when you're collaborating with scientists to be able to learn the science well enough to feel like you can really make a serious contribution to the field?
Oh, that's a very good question. So, of course, it depends upon the area, and it depends upon your aptitude. Like, early on in my career, I worked with a criminologist, and, he ended up getting really a lot of accolades for that work. And I helped him a lot in the beginning, and I could never get the hang of it. And if criminology's I don't mean to be insulting, but it seemed like it was easy. But I never had a knack for it, so it didn't suit me. So you actually have to think about, I lots of times work with Larry Wasserman, and Larry Wasserman is good at physics. Larry is not good at genetics, but he's a good collaborator because he has wonderful ideas, and I'm good at genetics. And I learned it over the years. So some things you can learn fast. You can usually learn enough to do something, and then you get to know more. You never know enough, though. You always have collaborators, and you listen to them. So I don't think you are never, really honestly, I'm always glad, even though there's advertising more broadly, I'm pretty glad that no genetics people walked in here to hear how I explained how CRISPR works, because it's not quite right. I mean, it's more than not quite, but it's close enough. And I honestly believe, a lot of people will make their graduate students take genetics classes. I never do that. I don't think you should know that much. If you know too much, then you'll lose your ability to model. Because modeling is capturing the big things and ignoring the little things and letting your collaborator tell you what are the big things.
Awesome.
Oops, this way. Yeah.
Oh.
And with the questions that you have on the list, when you're done with those, I'm going to try to ask the question without time.
Okay. So as a follow-up for the interdisciplinary research with other experts in other disciplines, so what are the strategies you would recommend to counteract how other scientists are often hesitant to use statistical approach and encourage further collaboration?
Let's see. So I think the question was that what do you do to encourage your collaborators to try more elaborate statistical procedures? Was that the gist of it? And when I collaborate with people, in the beginning, I generally I'm kind of old and lazy, so they have a postdoc or a student, and I guide that person via Zoom on how to do the easy things, the off-the-shelf things, as well as possible. And I always do that because if they haven't seen that, they'll never take the next step. They have to be convinced that there's a need for it. So let's say, they all know how to read those QQ plots. They look at that and they say, okay, I'm not very happy with that. I'm willing to try something different. So, seeing is believing, and that's how generally you do. People don't like to they don't like to be talked down to. So they want you to talk to them on their level, say, I see what you're saying. I agree it would be nice if it worked. It didn't work. Now let's take the next step.
So, thank you.
Maybe it's a good point
Yeah, if you want to.
question that I have no voice, so I need the microphone today.
Sometimes the opposite is true too. We are enamored with the things that we want to do, and there's no need for them, right? And actually an easier thing could give the result as well. So on this passage that you were talking about, that I think is quite critical, between doing things ad hoc that tell you what's actually the big message in the data, the strongest signals in the data, and then develop the new method afterwards, in a sense. Can you talk a little bit more about that? Because I think that's really critical for a lot of people here. How do you start by just, working with the data in a very ad hoc fashion, and then you take off and you build a new method?
So one of the things that's always a problem, I'm just going to give a little bit of background. So one of the things that's always a problem is that if you have a good collaborator, they are in a big hurry to publish, to get ahead of their competitors. And so I always am sympathetic to that, because otherwise they will boot you out and get somebody else. So I try to help them get that paper out, and I'm happy to do so if everything's valid, and the simple methods work. I'm completely happy with simple methods because I love science and I want science to move forward. That's my #1 goal. But then I have students and postdocs, and they have to have papers like in the Annals of Applied Statistics and stuff. So I always get the agreement. In the early phases of collaboration, I say, okay, we're going to help you do this, and we want the agreement that we can use your data in the next paper so that we can try our fancy pants stuff and see if it works. And they always say yes. Sometimes they want to be authors, sometimes not. It never matters, and it's all good. So I think it's all a matter of knowing. Like, I really get upset. This is just going to be my only scolding comment, is that sometimes people say all they did was get the data, and you think, oh my God, that took 5 years of postdoc work. All they did, they might say all you did was run R. They're all respectable things that are merit treating properly.
So it looks like we have time for one more question, and the question is, how important is the replication of results in an independent data set for single-cell RNA data, especially given the sparsity issue?
Well, now that's a very interesting question. The question is about, do you need to replicate it? And I've always been, I used to work in genetics, kind of migrated to genomics more recently. Genetics is such a perfect field. All the scientists know some genetics, and we can never publish without replicating. It's an amazing field. It's not like sociology or psychology, where they're always having to retract their papers. But unfortunately, in genomics, that stuff's expensive, and we're just starting to get it. So we are not replicating it, and I myself have had things that have not replicated. We all will, until we start to get enough data. But once we have enough data, we really should be replicating it.
Awesome. Well, thank you so much for answering all of the questions and for giving a wonderful talk today. If people have any further questions, there's going to be a reception on the 3rd floor of this building. So please, everybody come to the reception. And Merle, did you want to hand it back? Okay. Thank you.
Thank you.